All right, why don't we get started? You'll come up anyway. So do you want me to tell you when you've got like 5 minutes left? Do you want 30 minutes? You want 35? Okay, I think it'll be fun. I'll tell you when it's about 5 minutes, okay? All right, so we're going to get going because I don't want to have to take too much time out of the coffee break, which is what I promise I will do, because I will not cut short a minute of these two presentations because they're both going to be fabulous. And we actually tried to get this thing together for last year, and we weren't able to pull it off for a variety of reasons. And so, we had to invite Edmund and Anita to do it this year, and we're really excited about it. This is going to be really two talks that look at genetic and environmental risk factors for ADHD, and that they really, I've seen them both. I believe they do an excellent job of bridging the gap between science and clinical concerns. Certainly that is one of the main interests of Anita, and Edmund's opportunity to tell us about extreme environments and their relationship to ADHD will be of great interest to our group. Let me just introduce both of our speakers now so that I don't interrupt the flow as we go forward. So Anita Thapar, who told me that this is really her first presentation at a U.S. meeting, a U.S. psychiatric meeting, so that's really wonderful that we were able to get her here to do that. And you see the title of her talk up there, I won't repeat it. She is professor of child and adolescent psychiatry in the Division of Psychological Medicine and Clinical Neuroscience at the MRC Center for Neuropsychiatric Genetics and Genomics at Cardiff University School of Medicine. And our second presenter is Edmund Sonuga-Barke, and he will present Beyond Heritability, have studies of the effects of institutional deprivation change how we conceptualize ADHD. And Edmund is professor of developmental psychology, psychiatry, and neuroscience, and he wanted me to emphasize that the order is important. And he's at the Institute of Psychiatry at King's College, and he's also the editor-in-chief of Journal of Child Psychology and Psychiatry and whatever, related disorders. Anyway, there we are, and I'm going to turn the mic over to Anita, and we're very much looking forward to your presentation.
Okay. Well, thank you very much, Jeff, and thank you for inviting me to this meeting. It's a great honor for me to be presenting this morning. So Jeff gave me this title on, can you talk about genetics and environment? Now, I should let you know I'm somewhat famed for being able to talk very fast, but I thought even me, who can talk very fast, cannot cover the whole of genetics and environment in 30 minutes flat. So I had to give some consideration on what I was going to talk about this morning. So let me give you the context of where I started to think. So I come from Cardiff. This is where Cardiff is. It's about 2 hours from London. It's the capital of Wales. And this is the building I work in. You can kind of see that although some people say Cardiff is the wettest city, one of the wettest cities in the UK, the sun always shines in Cardiff. So, upstairs in our building, we have labs and research offices, and downstairs, we have the clinic. So I thought what I will do this morning is, because I cannot cover the whole of genetics and environment, I will take some of the genetic and environmental findings that are coming out from our own lab and many other people's labs around the world, and take those that I think about when I'm in clinic and share that with you. These are my disclosures. My research funding is from the UK Research Councils and from charities. And again, to stay focused, as you know, there are different types of genetic designs. I am going to focus on the findings that have come from the direct genomic assessments. Let me begin, first of all. Probably many of you know there's a paper that was now been deposited in bioarchives where common gene variant discovery in ADHD, there are at last some findings. It has lagged behind in terms of sample size, GWAS studies of schizophrenia, and that's because there are so many SNPs that we are testing, single nucleotide polymorphisms, we need very large sample size. And it's only through unprecedented international collaboration that we have a sample size now of around 20,000, and there are 12 genome hits. However, this morning I'm not going to talk about gene discoveries or genome hits. I'm going to talk about where I think about findings in relation to clinical settings. So what has molecular genetics taught us about the nature of ADHD? Well, GWAS, as well as being used to undertake gene discovery, we can also use GWAS data to compute composite measures of risk alleles associated with particular disorders. And that's something that's really grown in our field. So you can take a composite of common risk alleles that are found in a GWAS, known as polygenic risk scores, and take them from one sample, and they predict ADHD in another sample. So for example, polygenic risk scores, I can see Steve here, say from Steve's ADHD sample, and predict the Cardiff ADHD sample, distinguishes it from controls. Over the last few years, there's been many other methodological developments beyond polygenic risk scores to take some sort of composite of these common risk allele variants to look at genomic correlations between different disorders, including ADHD. However, just consider that these composite risk scores, polygenic risk scores, at present, because they're very dependent on the discovery sample size, have very low predictive power. So you're not going to be very excited when you look at the kind of correlation or the prediction. However, it does provide us with something like a biomarker, a useful indicator of genetic liability to address specific hypotheses. So we're going from GWAS, which is hypothesis-free for gene discovery, to actually get a composite measure to test specific hypotheses. So turning to ADHD as a concept, for many years that we've known that whilst in our clinical practice we need diagnoses because so many of our decisions are categorical. Do we use medication or not? Do we see this child in a specialist service or not? That's helpful. However, we've known for a long time from epidemiological studies that ADHD behaves as a trait. There isn't a kind of great level of ADHD. We arbitrarily have a cut point beyond which we call people affected, but in terms of adverse outcomes, those who fall just below the threshold are at much higher risk of adverse outcomes than those lower. So in this regard, it behaves a bit like, ADHD behaves a bit like hypertension in relation to blood pressure. So what does the genetics tell us? So this is an early paper from Joanna Martin, who's a PhD student in Cardiff and then has become a postdoc. She's doing her fellowship at the moment. And she took ADHD risk scores from patients versus controls and found that they predicted ADHD trait levels in a population cohort. So how high someone scored along this trait in the general population. Many other people have shown that same finding. And in that big GWAS study I was telling you, the international consortium, I was particularly struck because what they were able to do is take the genetic look at the genetic correlation between ADHD diagnosis with the correlation with ADHD trait risk alleles in another big consortium, and look how strikingly high that genetic correlation is. I mean, I was quite surprised. That is a very high correlation, genetic correlation of 0.954 between trait and diagnosis. This is Evie Sturgia-Cooley from Bristol. She looked at it the other way around. She looked at the population cohort, this is Alsbag, a birth cohort in the UK, and looked at risk alleles influencing how high people scored on a trait measure of ADHD. And she found that this distinguished between ADHD cases and controls. And even more surprisingly, within cases, found it predicted ADHD symptom score totals. So basically, epidemiological, twin, and now molecular genetic findings really suggest strongly that ADHD behaves as a dimension. And sometimes when I'm in clinic and I get, in the UK, you may know that we have quite strict criteria for diagnosis. And I have a family where, you know, the mother's concerned that the child has not met criteria for diagnosis. I find it very helpful drawing out that dimension and explaining how ADHD, we conceptualize it and how it behaves. The second issue I think about in my clinic when I think about that underlying trait is that hypertension, compared to when I was a medical student, that cut point for treatment has changed. It's lower now. So is our cut point changed? Is it going to change? Should it change? The third thing I think about is that for hypertension in the UK, guidelines adjust the treatment level depending on comorbidity. So for somebody with chronic renal disease, the threshold is lowered. Now, we don't do that for ADHD. We know that ADHD with comorbidity has a poorer prognosis, but we don't think about those contextual factors when thinking about the threshold. So these sorts of things sometimes I think about in clinic when I see someone who's sub-threshold, but they've actually got multiple other comorbidities. So the other question, again, most people in clinical practice will see that ADHD shows high concurrent overlap with other neurodevelopmental disorders. Now, we don't use DSM-5 in the UK, as you know, but actually I like the clustering of the DSM-5 neurodevelopmental disorders, partly because there's such a strong concurrent overlap, and of course these are all typically early onset disorders, early in development. Typically you've got a male excess and they're characterized by neurocognitive impairments. So to me they're a helpful clustering. So what about the genetics? What does that tell us about the clustering of neurodevelopmental disorders? So again, this is work by Joanna Martin. She took the common variant risk alleles, the polygenic risk scores, and again, as well as she found that not only do they predict ADHD trait levels in the general population, but they predicted cognitive variables, a trait measure of language, pragmatic language skills, and also social communication traits akin to autistic traits. So you've got the same common variants influencing multiple neurodevelopmental disorders, suggesting a neurodevelopmental spectrum. What I've talked so far only about common gene variants. Well, even before the kind of new genomic technologies, we're all familiar with the kind of rare syndromal forms of genetic forms of disorder. And we know that disorders such as tuberous sclerosis, 22q11 microdeletion, Fragile X, these individuals can present with ADHD, but they can present with a spectrum of other neurodevelopmental problems. We know that. More recent genomic findings in the last 5 to 10 years, people have been able to look across the genome at more subtle mutations. These are rare mutations, chromosomal deletions or duplications known as copy number variants, We knew very early on, as soon as the technologies came out, pretty quickly, that people with intellectual disability, including mild intellectual disability, have an elevated rate of these subtle chromosomal deletions and duplications. Interestingly, lots of studies have now shown an increased burden of these rare copy number variants in ADHD. What's interesting about these types of genetic variants is that they're rare, but unlike the common variants, they each have a large effect size. What's interesting is that these types of rare variants are also enriched in autism spectrum disorder, intellectual disability, also schizophrenia and epilepsy, but they're not so enriched and prominently enriched in mood disorders. So in terms of these findings, this really gives us a clue, again, about the neurodevelopmental nature of ADHD. The other interesting thing is that these CNVs for all these different neurodevelopmental disorders show significant overlap. So you've got the same CNVs resulting in different neurodevelopmental disorders. So ADHD seems to behave as a neurodevelopmental disorder, and that clustering does seem to work from a genetics perspective. But of course, if only research was so straightforward, there's a caveat. ADHD also shows genetic correlation with phenotypes outside that neurodevelopmental cluster. So these are findings from Lucy Riglin. For many years, we've known that emotional dysregulation, irritability, in fact, historically has been quite an integral part of ADHD. And Lucy found that ADHD polygenic risk scores predicted irritability in 2 population cohorts and a clinical sample. So that's one finding. This isn't a neurodevelopmental disorder. The chronic irritability is conceptualized as a mood disorder. The other thing is that ADHD is correlated with multiple psychiatric disorders, particularly depression. You've got a genetic correlation, and also with education, years of schooling. So this could be pleiotropy. You've got the same genes influencing multiple brain phenotypes, but that's not terribly helpful clinically. That's okay. And what really we're getting now in the next 5 years is new methods, genetic methods to test to what extent are the genetic correlations actually representing that this perhaps is a spectrum of ADHD, or that the relationship in terms of genetic correlations is causal. So, for example, it could be, it's plausible that ADHD results. It has a causal effect on years of schooling, or perhaps has a causal effect on depression. However, you can also have misclassification. We were talking last night over a glass of wine to somebody about maybe a lot of people with recurrent depression who are in the depression GWAS actually have undetected ADHD. So these are all really important questions that need to be resolved for the clinician. So I'm going to talk about ADHD persistence next. So we've known for many years now that ADHD is not childhood limited. It can persist in a substantial number of affected individuals, but not everyone. And we've again known for some time from family and twin studies that the genetic burden is likely to be increased, or at least the familial loading is higher in those who have a persistent picture of ADHD. So let's turn to the molecular genomic findings. So this is again work conducted in the Alsbach UK birth cohort, and this is Lucy Riglin, a postdoc. Now, Lucy looked at ADHD symptom scores in this population cohort using the SDQ, which has been, the SDQ has been basically administered to this large population cohort repeatedly. So we've got repeated measures of ADHD traits from age 4 to age 17. She then basically looked at the kind of developmental trajectories of this population. So the majority, because it's a general population cohort, just stay on a low symptom trajectory. You've got a group that perhaps we would be seeing in clinic who have got a persistently elevated trajectory. There's an intermediate group, and then you have a group that they start off very high and then they reduce, what we would call a childhood limited or desistant class. So Lucy looked at the ADHD polygenic risk scores and found that that persistent group had a higher burden of polygenic risk scores, not just more than the low and the intermediate, but interestingly, more so than the desistant. It distinguished the persistent from the desistant. Now, because I've talked about pleiotropy, you might think that other polygenic risk scores would predict persistence of ADHD. And in fact, I work with a lot of the colleagues in my own department work a lot on schizophrenia genetics. And the schizophrenia gene discovery sample is huge compared to ADHD. And my colleague Mick said, bet you schizophrenia will be better at predicting ADHD persistence. And it did not, nor did bipolar, nor did mood disorder, even though they're much better powered than the ADHD risk alleles. So I just want to highlight, everything is not non-specific in genetics. This was ADHD polygenic risk scores. So that's interesting, Lucy, but I don't have polygenic risk scores in my clinic, so have we got some sort of marker, apart from family history, which isn't always easy because parents don't know whether they've had ADHD, at least in the UK, have we got some sort of marker? Now, we thought, given pleiotropy with and a neurodevelopmental spectrum, perhaps those with higher ADHD polygenic risk scores would have multiple neurodevelopmental problems. And I'm really interested in this group because that's a lot of the children I see have got those difficulties. So we wanted to test whether neurodevelopmental multimorbidity, not just having ADHD, but having other neurodevelopmental problems. So we looked at language problems, lower IQ, social communication autistic traits, and actually we included conduct problems here. And we found that the ADHD polygenic risk score loading was associated with a number of with a degree of multimorbidity. And those with more than 3 neurodevelopmental problems had higher genetic risk scores. So it did appear to be some sort of clinical indicator. And the multimorbidity levels differed across those 4 trajectories. It didn't explain all the polygenic risk score effect, but still, it gave at least some sort of clinical indicator of persistence. So I can't talk about genetics to a mainly clinical audience without raising this issue about genetic testing. And certainly when I see psychiatric trainees, I'm giving a talk on genetics, this is all very nice, but it's not going to mean anything to me clinically. We're not going to ever have genetic testing in child psychiatry. I don't really have to concentrate on what you're telling me. Well, common variants, the polygenic risk scores, as I said, they're nice for testing hypothesis, but they are weak. We cannot use them in clinic. That's not funny about ADHD at the moment. We're in that position for other common complex disorders. However, does that mean we can just forget about genetic testing? Well, I just want to raise the issue about rare mutations. Now, I've told you about rare mutations that individually have large effect size. And there are ways of being able to identify which ones are likely to be causal, for example, looking at de novo mutations. And some of them we know are medically actionable. They have clinical implications. So what sorts of clinical implications? You might think, well, I don't know, is it really relevant to me in my clinic? This, I think, is a very interesting example. This is a colleague from Exeter, Andrew Hattersley. I think he's a very modest man, but he's done really nice work on diabetes. So he's been looking at type 1 diabetes, and particularly in those who've had neonatal diabetes, which typically has persisted, but it might have desisted and then restarted, and found there's been 2 rare mutations involving the pancreatic K-ATP channel. So this should be 2004, I believe. And what was interesting is from that basic science, he worked out the mechanism that this group
would be likely not to respond to insulin, which is what would be routine clinical practice. They would respond to sulfonylureas. And actually, papers since then have suggested that this group do not, they show not a very good response to insulin, and they show a good response to sulfonylureas. And I think it's very impressive. He, with a Wellcome Trust, are actually trying to set up global, a mechanism whereby even if people come from no matter what country, they can get tested here. So this is an example where it's made a difference to treatment, and particularly in early onset. So what about in psychiatry? Well, we're already at the stage of screening in intellectual disability. Now, I first became a psychiatrist in 1986. And when I first trained, we thought of intellectual disability severe. That's genetic or environmental insults. Those are the children you send to your clinical genetics colleagues. Mild intellectual disability, sociocultural. And we learnt that for years. And now with these different rare mutations and increasing number of copy number variants being found, we now don't think anything about screening for mild intellectual disability. And in the UK, we have universal screening now for kids with developmental delay, mild intellectual disability. And for autism, I know you have that in the US, we don't have that in the UK. So people who know me will know I'm quite cautious, but I will tell my trainees that I do think it's not going to be very long before we screen for these sorts of mutations for the other neurodevelopmental disorders, not just intellectual disability, also ADHD, and I'm guessing probably schizophrenia as well. Because for some people, it could have consequences for treatment, picking up something medical, and maybe even very early intervention at the neonatal stage where it looks clinically the same, but it's not. So I've now also got to cover environmental risk factors. Now, no psychiatric disorder is entirely genetic. We know that. And wouldn't it be cool to find environmental factors? But I just want to kind of highlight that I actually started off, before going into genetics, interested in environment, and then I switched. It is actually a real challenge to find out what's causal. So we know that for ADHD, it's an early onset disorder, so the life sort of risk factors we're thinking about are early exposures. Of course, it doesn't mean that later exposures aren't important, but they're more likely to be important in terms of modifying the clinical presentation or the outcomes. So this is Kate Langley from Cardiff. Kate's done a lot of work on this. These are the sorts of risk factors we know are associated. Whoops. The problem is, and Brian talked about this yesterday, if we observe association in case control or any sorts of longitudinal cohort designs, which I like, as you know, association does not mean causation, because there are many other reasons that risk factors and clinical outcome can go together, amongst them reverse causation and confounding factors. And you really need quasi-experimental designs that pull apart the confounders and other sorts of unmeasured risk factors. And there's been loads of research showing even if you think you've got measured covariates, that does not do the job. So let me give you one illustrative example. Maternal smoking in pregnancy, which was my favorite risk factor in the 90s. What do you think? It could cause offspring ADHD through nicotine exposure, carbon monoxide, and disrupted placental blood flow. However, we know now from GWAS that smoking, and including maternal smoking in pregnancy, where you can't give up, is strongly genetic. So as well as familial confounds, you've got potentially maternal genes here, maternal genes being transmitted to the child, a confound which you won't necessarily be able to measure. Ideally, we'd like to remove the genetic link. One way we did that in the early 2000s is by looking at children born by IVF, where some are genetically unrelated to the mother and some are related. These are my colleagues, Gordon Harold from Sussex, Francis Rice from Cardiff, we did that study with. What can we test? If maternal smoking in pregnancy has real risk effects on the child, it won't matter if the child is related or unrelated to the woman undergoing the pregnancy. And it didn't for birth weight. Smoking in pregnancy is not good for offspring. However, the related, we can see an association, just like in any observational study, but nothing in the unrelated. But, you know, we're skeptical, lots of problems with this design, but there are many other designs. I can't go through them all, but like, you can look at mums where they smoked in pregnancy and dads, and if it's a genetic link, you'll also see association with paternal exposures. Brian and others have looked at siblings where mum has smoked for one pregnancy and not the other, and shown elevated rates of ADHD in the unexposed. There are many different informative designs, and actually Frances Rice has just done a very painstaking systematic review. It's in press. She's found 12 informative studies, and basically the conclusion, when you look at informative studies, it looks like maternal smoking, although it's a bad thing, for ADHD is a public health measure to reduce ADHD. Maybe we need to be looking at other environmental risk factors. Interestingly, looking at the molecular genetics, my colleagues in Bristol found that the ADHD polygenic risk scores predict these prenatal exposures. So, just to wrap up, ADHD we've known for some time, it's strongly heritable. The architecture, like all complex disorders, they have common variants and rare gene mutations. I want to say that the genetics works with the epidemiology, it behaves as a trait, and that's something for us to think about in conceptualization. There's a neurodevelopmental overlap in terms of shared genes, and the ADHD persistence, they have a higher genetic burden, look out for multimorbidity. Genetic testing in clinical settings isn't here, but I think really look out for practice changes in the future. Identifying environmental risks is important, but I just want to highlight it's really a challenge to identify what's causal. That's almost the first thing I say to medical students who read a case control study, see someone in clinic, say, I know why that mother's child's got ADHD. Just be careful. Is the environmental factor an index of parent or offspring genetic risk or some other variable? Causality needs rigorous testing, and I think it's great Edmund's going next, because Edmund is going to describe a quasi-experimental design which does exactly that. And I just want to highlight, genetic designs aren't just for gene discovery. They can elucidate environmental risk factors, causal environment, not just genes, and test causal hypotheses that are clinically relevant. Thank you.
I'm afraid if I invite one or two questions, I'm going to open up the floodgates, because this was a very, very rich and engaging presentation. But Edmund says we can take one or two, but that's all. And oh, that's why he said, by the way.
Brilliant, Anita. Is this on? As always. Yeah, so we want to design a study of at-risk kids. Now, very often, typically, in the past, we've looked at familiality as a marker at risk for ADHD, so having relations with ADHD. Compared to that, would polygenic risk scores provide a better, more powerful marker of risk in those sorts of designs? So if we screened, did a GWAS, and looked at the polygenic risk score for ADHD, would it be stronger than just saying taking kids who've got a brother or sister with ADHD?
Okay, that's interesting. I think you've taken the right approach. Family history is still stronger. But something to look out for is, I mean, there was a paper that it came out a couple of years ago. It wasn't about psychiatry, it was about common complex diseases, about combining family history with polygenic risk scores. And I think that's something that we have to look out for as the sample sizes for GWAS discovery increase. I think polygenic risk score on its own is not. I would go for family history, but I think that's an empirical question. But for the moment, yeah, stick with family history.
Thank you.
But look to the future.
Tony, Russ is first.
Yes.
And I'm cutting it off after that.
Okay, thanks. Anita,
Tony, hold it for the discussion, okay? Last question.
Great.
I know, he snuck in there. What can I tell you? Go ahead.
Are we ready? Okay. One question I have is, in the area of autism, de novo mutations now appear to be playing a much larger role, perhaps in some studies accounting for 25% of all new cases in which there was no prior history in the family, but obviously with parents waiting longer and the risk to mutations particularly being 3 times more likely in the fathers than the mothers who are delaying their parenting is now contributing to some extent to a rise in ASD. And I did see one paper that suggested that that might also be the case in a small percentage of ADHD cases. What are you finding in your research about the role of these de novo, newly arising mutations in children that are not actually present in the parent's genome other than in their gametes? So.
I think that's a really important question, and it's a question that makes me slightly sad because the de novo mutation research is, obviously there's a lot been done in intellectual disability, loads done in autism, stuff done in schizophrenia, and this is like, it's shameful. Where is ADHD in all this? And I don't think it's for want of researchers not wanting to do it. I think it's just one of those things, it's not viewed as sort of a top priority. However, we don't have that much published data, but certainly this is hearsay now, so it's not published. But I have heard that a large sequencing study looking at de novo mutations in ADHD surprised some people because guess what? They find a rate of de novo mutations pretty similar, if not a bit more than ADHD, than autism. Yes. So yeah, I think I'm sad that it's lagged behind because it ought not to have, but I think that will change. So I think it's definitely worth looking out for the papers, and I'm really glad you brought it up.
Okay, thanks. I look forward to it. Thanks, Anita.
Oh God, it's really complicated. It's
Tony, I promise you can lead off first question in the discussion period. Edmund, the mic is yours. Oh, you gotta get wired?
I'm just getting undressed on stage.
Hello?
Stick it on your bow tie.
Oh yeah, it's working. Thank you so much. Yeah. Try and find a space for it. How's that? Okay? Great. Well, thank you so much for inviting me to speak in this plenary session, and wonderful to share the stage with Anita. Thanks in particular, of course, to the committee, to Mark, to Jeff, to Tony, for this honour, and for also giving me the opportunity to come to APSARD for the first time. Intellectually very stimulating and also a lot of fun, so that's great. So DSM-5, like its immediate predecessors, adopts a phenomenological approach to defining disorder based on manifest symptoms and impairment. But of course, not far below the surface, those concepts are underpinned by assumptions about etiology and cause. And we can see those when we read papers, or even when we write papers, we can see our own assumptions in that first 2 or 3 sentences in a paper. You know those sentences? ADHD is. Cut and paste, you know. And although I've not done a textual analysis of those little first paragraphs, I'm sure the vast majority mention genetics in a more or less sophisticated way, but even to the extent of some saying ADHD is a genetic disorder. So today I'm going to push up against that a little bit, and first of all I'm going to ask the question whether ADHD can be non-genetic in its etiology, so that it can be present even in individuals with low genetic liability. Secondly, in those cases, where do they fit into the kind of heterogeneous and polythetic construct of ADHD? Can it cope with those cases? And thirdly, what are the implications for nosology and taxonomy of those cases? And I'm going to speak to a number of headings. First of all, can extraordinary environments have extreme neuroplastic effects that override genetic effects? So introducing the concept of extraordinary environments. Early institutional deprivation as an example of an extraordinary environment, and its links with ADHD, and in this case particularly adult ADHD. I'm going to introduce to you a test, a quasi-experimental test of the role of those extraordinary environments in the emergence of ADHD, the English and Romanian adoptees study, and in this case the brain imaging aspect of the study, the arm of the study. I'm going to look at the effects of early deprivation, in particular on neuropsychological functioning. It's the first set of data we've really analysed properly, so I'm going to focus in on that. And ask the question, do deprivation-related ADHD cases differ from typical clinical adult ADHD cases in their neuropsychological profile? To try and get at those issues of the equivalence of deprivation-driven and non-deprivation-driven ADHD. So, traditionally, of course, ADHD is and has been identified as a highly heritable trait. And this has pushed the focus onto genetic factors in etiology rather than environmental factors. But however, given the observational nature of most twin studies generally, those heritability estimates depend on the type of experiences that the studied population is exposed to. And you could call these normative experiences within normative populations. They say very little about the potential of extraordinary environments or rare environments to determine or cause disorder. So while genes are strongly implicated in normative environments, extraordinary environments may override those genetic effects to either provoke disorder, even in otherwise strong, invulnerable brains, or prevent or remediate disorder in vulnerable brains, genetically vulnerable brains. So we can think about this in terms of neuroplasticity. So normative environments, of course, interact with genetic factors to mold the mammalian brain in complex and subtle ways, to bring about learning and development. That's one sense of neuroplasticity. And within the literature, that's the sense that's used most within the neurosciences. But then we talk about neuroplasticity in a much bigger sense. If we move beyond those normative environments, having those subtle effects, can they produce extreme neuroplastic responses that have profound consequences for development? And that's an area we really know very little about, actually. So we can think of neuroplasticity in this sense as a double-edged sword. On the one hand, we can ask, can adverse environments do such bad that they can derail development in an otherwise healthy brain, or in a brain of an individual at low genetic risk? And the example there is the Romanian adoptees that I'm going to talk about. And on the other hand, can an enriched environment do such good that it can put development back on track in an individual's brain who's at high risk of high genetic risk for disorder? Double-edged nature of that sword. So we're going to focus on the negative side of that sword. And the skeptics in the audience will say, and I'm the most skeptical person probably here in general, is sure, in principle, this seems possible, but in reality, where's the evidence that environments can have such a powerful effect? Well, this is the focus of the talk today. This is the the example of extreme adversity that was experienced by these young people in the orphanages of the Romanian state at the time, around the time of the fall of the Ceausescu regime in the late 1980s, early 1990s. And people would probably have seen, if they're of a certain age, they'll have seen those pictures on the telly of those filthy and emaciated children staring back blankly at the cameras. In terms of the exposure, it's a complex exposure, but it's certainly extreme. Severely restricted diet, high rates of communicable diseases, little or no social and cognitive stimulation, and no personalized care, no chance to establish those selective bonds or attachments with adults. So they lacked a range of early childhood experiences which we assume are essential for normal human development. So it's a very powerful test of the effects of environmental adversity. But what was the effect? So I'm the principal investigator currently of the English and Romanian adoptee study, a study that was established by Professor Sir Michael Rutter at the request of the British government back in the early 1990s. And it's, I think, a unique natural experiment of the effects of early experience on development, involving the adoption of profoundly deprived infants from those Romanian orphanages. So they spent between 1 and 43 months in the orphanages, usually from around the first 2 weeks of life. So there wasn't much pre-institutional exposure going on, except, of course, prenatally. Then they were adopted into loving, caring, well-functioning families. And we've now followed them up to 25 years. So we've gone into young adult period. And the crucial thing in terms of Anita's point is that the duration of the deprivation is probably unconfounded by genetic liability or liability to other exposures. And we can talk about why that's the case, but we actually have polygenic risk scores, and we're looking at correlations between the exposures and those risk scores. So that's the design of the study. And we followed them now up at 4, 6, 11, 15, and now young adulthood. And in 2017, last year, we published a paper in The Lancet where we described the developmental trajectories of all those families, all those children, rather, in those new adopted families. And what we found was, when we compared those with over 6 months deprivation, compared to those with under 6 months deprivation, and then against a control group of UK adoptees who were adopted before 6 months and with no evidence of deprivation, we found initial devastating effect of the deprivation, as you can see from this graph here. Does the pointer work? No, it doesn't. This is the. Is there one that works? This will work. Better. Oh yeah.
It won't
Thanks.
go to both. You just have to...
So this is the proportion of kids with what we call cognitive impairment. We defined it as IQ of under 80. So you can see the red group with the over 6 months group, and this is what it's going to be in the rest of this slide. Over 6 months group had almost 50% had cognitive impairment by that definition. The under 6 month and the UK control group hardly differed. This was by the age of 6 years. But then you can see quite a remarkable recovery in the IQ levels, or a remission of cognitive impairment in those over 6 months as they grew into young adulthood. That's the first really important part of the developmental phenotype in this group, recovery in the adoptive families of cognitive function. However, when we look at neurodevelopmental problems, we don't see a similar pattern of recovery. So levels of quasi-autism, as Mike Rutter termed it in those early papers, autistic symptoms, remain just as high right the way through to young adulthood. Levels of disinhibited social behavior, which of course is very characteristic of children who've lived in these sorts of institutions, remains just as high. The effect is just as large whether you look at 4 years or young adulthood. The levels of inattention overactivity on the Rutter scale and the equivalents, clearly a marker of ADHD symptoms, remains just as high and even may get stronger as you get away from the initial exposure. It's really quite striking. So from a clinical perspective, the big question is whether those patterns of those patterns for inattention overactivity are also found or reflected at a diagnostic level. So we did diagnoses, we did a diagnostic workup at both adolescence and young adulthood, and we published this data in the JCPP in 2016. And what we can see is, the answer of that question is yes. So a very high proportion of our young people with over 6 months deprivation, that's these two groups compared to these under 6 in UK, meet the diagnostic criteria for ADHD. Talking 30 to 35%. And this is in young adulthood. And we used a very, very strict criteria. We actually used the childhood DSM-4 criteria rather than the DSM-5 adult criteria for this. So this is symptomatic and impairment. So really very strong persistence, even escalation of the problems. And when you compare that with the age 15, you can see that escalation. Very striking. So a much smaller proportion of cases using the same definition met the criteria at 15 compared with at young adulthood. So the actual risk ratio at 15 was 1 to 4 times more likely in the over 6 months group than in the combined control group, compared with almost 8 times more likely in young adulthood. So it's like an escalation of ADHD problems in this group. Really quite a different pattern you see in a normal clinical sample. In terms of the sex ratios, we see, and we always have seen, no sex differences in our sample. One way that this differs from the typical clinical presentation. In terms of the presentation, we see, and we always have seen, a predominance of inattention rather than hyperactive impulsivity. So the majority have an inattentive presentation and always have right the way from the early years. So some things that are quite specific, but still meeting the criteria for ADHD. How should we think about that? That's the question I'm putting to you in a way, as an audience. In terms of comorbidities, an unusual pattern of comorbidities. You've already seen the high levels of disinhibited social behavior and autism in the sample, and not surprisingly, those co-occur with the ADHD symptoms. Impairment and treatment. I mean, the very striking thing in terms of impairment, in particular, if you look at the very high rates of unemployment in the extremely deprived kids with ADHD compared with the other groups. That's really right up there. Nearly every one of those children in that group unemployed at the time of this study. And you've got to remember they were adopted into high-functioning, relatively wealthy families with brothers and sisters who are probably lawyers, accountants, and doing really well. That's quite a risky context, I think, for the development of mental health problems. In terms of medication use, yeah, a lot have received medication over the time, for ADHD specifically, so stimulants or non-stimulant ADHD medications. So in the over 6 months group with ADHD, you can see about 40% have received treatment for ADHD. So as far as the doctors are saying, they're saying this is ADHD. But how typical, more generally, are those cases of ADHD? And in particular, focusing on the neuropsychological presentation of those different groups, compared with common or garden clinical ADHD. This is a work in progress, so no photos at the moment. It's not published yet, but I thought it'd be good to give you a heads up on what we're finding. So there's a picture of the ERA team, particularly related to the ERA brain imaging study team. There's Mike in the middle, of course, where he absolutely should be. He's the inspiration for the study, and still is our chair of our steering committee, and he still makes a great input. Dennis and Nuria, two of the research fellows, are really focused on the brain imaging stuff, and Mittal Mehta's really leading that. He's a colleague at King's College London. So we focus on 3 brain networks, And we identified these networks, candidate networks, for two reasons. First of all, animal studies in particular show each of them as being affected by early stress or adversity in the rat models and the monkey models. And that's the dorsal frontostriatal executive system, the ventral frontostriatal reward system, and then the limbic system, of course, highly sensitive to early stress, involving the amygdala and projections across the brain. So those are our three sort of candidate systems, and we developed our protocol around those systems. In terms of the design of the whole of the Erebus study, the brain imaging study, we've got obviously structural work on voxel-based and surface-based morphometric parameters. We've got brain function, and we're looking particularly at inhibition, reward sensitivity, empathy, the empathy accuracy task, and emotion processing. So we had a limited amount of time. We could have, we had a big arguments about what we should put in. That's what we could put in. But they map onto our brain networks. Connectivity, of course, and those data are coming through and very exciting, both in terms of structural connectivity and the white matter tract integrity, and resting state connectivity. So we have a resting scan. And then neuropsychology, which I'm going to focus on today. We look at inhibition in a go-no-go task, risk-taking, the risky choice task, prospective memory, and emotional processing. And our numbers, this is always a frustration for us, of course, even though this is by far the largest sample and most homogeneous sample of severely exposed kids in the world. You're always struggling against numbers. And so you can see we have 60 Romanian adoptees, we have 23 control UK adoptees, we have a non-adopted healthy control sample we've just recruited, and we have a clinic-referred ADHD and a clinic-referred autistic adult sample. We have, then we looked, of course, at attrition, and in general, there isn't selective attrition, but there is an underrepresentation, perhaps, of kids meeting ADHD at the young adult follow-up, which is something we've got to think about what that means. So in terms of the inhibitory control task, we actually focus on this concept of proactive inhibition. That is a sort of state of tonic readiness to inhibit rather than the reactive inhibition, kind of phasic response to an ongoing stimulus. Because there's a lot of methodological work showing that those two processes are kind of confounded in most tasks. So we use a queued go-no-go task, where we queue whether there is going to be a go signal definitely coming up with a blue cross, for instance, or whether with a red cross there's a 50% chance you're going to have to stop. And then you look at proactive inhibition by taking the red from the blue cross trials, if you like, that difference between when you're prepared to stop and when you're not, when you're prepared to just go. Prospective memory, we all know what that is. That's remembering to do a future task when invited to or at a pre-specified time, uninvited. So we're looking forward. Somebody says, in 10 minutes, please remind me to do this, or please press that button, or please write your name down on a piece of paper. The two aspects to that, prospective memory, there's a kind of spontaneous aspect of prospective memory, but also an intentional aspect. The intentional requires more monitoring and maintenance than the spontaneous response. And we measured that by a test called the MIST, Memory for Intentions test. And it's a series of tasks. It's used in the diagnosis of Alzheimer's or dementia, actually. And it's a series of tasks where the patient, the subject is, the participant is asked, for instance, in 15 minutes, tell me that it is time to take a break, for instance. Or when I hand you a red pen, put it behind your left ear, or whatever. Yeah? So some are time-based and some are instruction-based. Risk-taking, we used a risky choice task, and crucially here, in this task we developed at Cambridge by my colleague Graham Fairchild, the contingencies are completely transparent. There's no need to learn the contingencies. They're presented with little wheels of fortune. They have to decide which one they want to go with, and the chance of reward and the probability of reward on each of those two sides is actually completely explicit to the individuals. And crucially, we actually look for risk-beneficial responses and risk-detrimental responses. Very often, again, in risk-taking studies, the risky choice is always detrimental. We didn't look at that. We wanted to try and disentangle that dysfunctional behavior from actual risk-taking. Emotion recognition is kind of easy, so I won't dwell on that. Presented with a series of emotional faces, have to categorize them. And a control task of categorizing by age. So in terms of a neurodevelopmental context, just a little bit of brain imaging data. So here's the total grey and white matter volume of our three groups. Our controls, which is a combination of healthy controls, and our non-Romanian adoptees, our adoptive control group. Our ADHD group, who came from the clinics, and our deprived ADHD group. And you can see that there's a severe stunting still in young adulthood. We're talking young adulthood here. So after 25 years, there has been catch-up, for sure, but still severely stunted in terms of the overall brain size. Interesting, not particularly highly correlated with general physical stunting and heightened growth, which surprised us, but that apparently is the case in general. In terms of IQ, we still have significantly lower IQ in our group. Again, that's a part of the phenotype. It really raises this question, do you control for IQ or not, when IQ is a fundamental part of the outcome of an exposure. So what about the effects? So what's the general effects of deprivation? The first of the effects of adoption. So we've got a healthy control group in. The reason we put those guys in was to try and rule out the fact that adoption itself could be having a problem. We could be creating problems. Because, of course, we have a UK adoptive group, which was our control group, but clearly, if you haven't got a normal, healthy control group, that may be underestimating the effects. But what we actually find across all of our tasks is very little difference between our healthy control group and our non-Romanian adoptees group. So there doesn't seem to be effects of adoption on neuropsychological functioning in general, although there is some apparent effect on proactive inhibition with reduced levels of proactive inhibition in our adoptive group. So what about the effects of deprivation? Clearly this is our main focus and has been for the last 20 years. We see a very strong effect of prospective memory, as you can see from this graph. So the green is the control group, that's the combined control group, UK and UK adoptees and healthy controls. Then we have the under 6 months and the over 6 months. But crucially, and very differently from nearly all of our other studies, there's a risk of simply being from an institution, irrespective of how long you were in that institution. So there is a big effect, even in the under 6 months group. The role of IQ is interesting here, and that doesn't, even if you control for IQ, which I'm not quite sure we know whether we should or we shouldn't, there is still an effect. In terms of risk-taking, we find that, in fact, the control group make more risky choices in the risk-favorable condition, where it's actually beneficial to make risky choices. Okay? Our Romanian group made less risky choices in that situation, but if you look at the risk detrimental or unfavorable, you can see the opposite pattern that is not quite significant. So they make more risky choices when it's detrimental, but less when it's favorable. So they're kind of just bad at making decisions. It's not like they're really big risk takers, they're just not very good at making those decisions. Emotion recognition, and here's a big one. So there are big deficits in emotion recognition in terms of the percentage correct Nothing more sophisticated than that at the moment, but you can see particularly at the negative emotions, sadness and anger being the biggest effects, but overall, you can see overall there's a large effect, but again, independent of duration of deprivation, and we've not really seen this before, at least certainly not in the late childhood and adolescent follow-ups. There's always been that dose relationship between deprivation and outcome. In terms of proactive inhibition, what we find is reduced proactive inhibition in the more deprived children, but not quite reaching significance. So that's the index of proactive inhibition. You can see reduced inhibition in the over 6 months group. That seems to be quite specific to that group. So what about the comparison between ADHD in the deprived group and in our clinic sample? So here's the controls to ADHD in general. So we can collapse the two groups in this analysis, and you can see a big effect, impaired prospective memory when we do that. This poor decision making, you can see less risky choices when they should have been making risky choices and more risky choices when they shouldn't have been making risky choices. But again, not quite significant. That's the dotted line. I think it's 0.06. P. There's no effects on emotion recognition when you look at the group as a whole. So ADHD kids as a whole didn't have problems recognizing those emotional faces. And an effect, a marginal effect on proactive inhibition. So reduced proactive inhibition in our ADHD cases. So reduced kind of tonic readiness to inhibit. Okay, so that's the effect of ADHD as a whole. What about the effect of deprivation-related ADHD compared with typical clinic-based ADHD, whatever that means? Prospective memory, it's the deprived kids, so ADHD plus deprivation plus D, that show the largest impairment. We have a power issue, but it's clearly more impaired than our clinic ADHD cases in prospective memory. Similar pattern, again, there's a power, we have this old power problem that we have, but similar, the clearest effect is with the ADHD kids from the deprivation group. In terms of emotion recognition, and this is the clearest pattern, I think, that the emotional deficit, the recognition deficits are really clearly only expressed in the ADHD kids who've suffered deprivation, not in the clinic. Say kids, these are young adults, sorry. Young adults who've suffered deprivation, not in the clinic sample. And again, in terms of proactive inhibition, the clearest effect is with the deprived kids compared with the non-deprived ADHD young people. Although, again, they are both, the pattern is that they're both different from the control group. So in terms of a discussion, call it a discussion rather than conclusion, ADHD is common in adults who experience institutional deprivation. I mean, really, really common. 8 times more likely than in our control group. Highly persistent, and that may be a really important characteristic of this form of ADHD, its persistence and its stubbornness. Individuals who experienced extended deprivation showed weakened prospective memory, slower information processing, reduced proactive inhibitory control, impaired processing of negative emotions, and altered decision-making in the face of risk. ADHD in general was associated with impairments on prospective memory and proactive inhibition, largely independent of deprivation status, although the impairment seemed larger or greater in the deprived group. But emotion recognition in particular may be a specific correlate of ADHD following deprivation. That may be, of course, due to the pattern of comorbidities that these individuals present with. Okay, thank you very much.
we'll have about 15 minutes for this.
So I was going to ask a question for Anita, but
Go ahead.
just as a comment to you, Ed, this is really, I was hoping for greater outcomes when you showed us that the IQ had normalized.
Yeah.
But then it kept dawning on me that, you know, so much else is going on and you're trying to pursue that and understand it. And the high unemployment, and just to me, that's probably shows that, are they getting disability of some kind? What's happening to them? And my main question is...
We'll ask Anita. Go on.
Okay.
Go for Anita.
So, well, actually, my question relates to both of you because I think we're now getting to the point where we need to measure the environment type. And sort of how do we measure the environmental risk in an environment type standardized way? I mean, we've been thinking about this a lot, but you provoked a lot of great thoughts in my mind about how if you look at the dimensionality of ADHD and those that live in more precarious risk environments, should we be not thinking about those interactions then in a different way rather than trying to standardize a single score on a single diagnostic test, that if we're looking especially at prevention, that those kids that are growing up in greater at-risk environments who are already showing even sub-threshold ADD ought to be given some help. But what we have now is a system that, in essence, tries to say, well, you don't have enough of these symptoms to get the help. And so I was thinking about this idea of how do you were challenging us to think about hypertension levels being pegged differently in England if you have renal disease. How might you imagine us doing that for these neurodevelopmental, in particular ADHD? Can we begin to measure environment type and add that into our diagnostic and intervention schemes?
Okay, I think I can think of about 3 different issues related to that question. I think the first is, I mean, I'm guessing that most of us here are child psychiatrists, pediatricians, or neurologists. I actually think that if ADHD and other psychiatric problems, some of them behave as trait measures, should we, in the way that people would think about blood pressure and hypertension, be talking more to our public health colleagues? Yeah? This is kind of like sort of outside our realm. I mean, it's not, it's our realm, but it's not, we're dealing with individual patients. So I actually think that these sorts of things need to become part of public health. And I've certainly, my colleagues in the UK, I've highlighted that, you know, a lot. I mean, the empirical evidence as to what you do is not there. The second issue is about stratifying in terms of comorbidities or contexts. Again, I think that's an interesting issue and, you know, one that I shared with you that I think about. Again, we need empirical evidence. The third thing is, and that's away from the clinic, is that I think in actually testing causal environments which may have important clues about what sorts of public health measures, we need to be rigorous. I mean, there's full of, you know, any public health physician will tell you about the sorts of stuff that, you know, has been wasted over the years for ischemic heart disease prevention, etc., etc. We don't want that in mental health. So I do think we need to turn to much more sophisticated epidemiological methods and our epidemiology colleagues to really try and find out the sorts of factors that are causal. And I would add to that is that, I mean, I really like, well, Edmund knows I really like his presentation because it does strike me about these rare environments. And we of course, we know now in genetics that you can have sort of rare mutations. And of course, these epidemiological methods, which I like very much, are great for looking at common factors. But they're not good designs for finding those rare situations. And perhaps some of the environmental risk factors, are we missing them because there are multiple types of lots of rare types of environments which we would not pick up in large population studies? So I raise these as questions. I'm sorry, I haven't given you any answers, but there's a lot of research to do.
I think, that's brilliant, of course, but I do think that one of the real key things, at least from the Romanian study, is that this is ADHD, this is an attachment disorder, you know, and so I think there's a tendency for clinicians, at least in the UK, if people have had a traumatic and adversity-rich childhood is to dismiss ADHD as a possible diagnosis, but actually in our case that simply isn't true. So I think that's the kind of clinical message from this, is that the environment can really drive a lot of the problems and shouldn't just be dismissed or shouldn't be misdiagnosed as some other sort of social relationship type disorder.
And I think that although you could not necessarily equate inner city urban life in the US or Europe with those extreme environments, I think it probably is very similar.
I can see there's a long queue, but I'm just going to say, I mean, it's really interesting. So people do often ask, and maybe one of the people in the queue is going to ask, in how relevant is this to ordinary patterns of deprivation? But actually, there is a study in England, it's called the Social Study, and it's by Jonathan Green up in Manchester, and it's with fostered kids like those sorts of kids who've been exposed to problems within their families and so forth, inner city areas, and they have a very similar profile clinically in terms of comorbidities with autistic type problems, disinhibited social behaviors. So this may be the extreme, but like most of the things, same as genetic factors, is a dimensional thing probably. So I do think there is a relevance. Sorry, yeah.
That's good.
That's good. In the deprived ADHD people who are on medication, do you know if there's any, is the response to the stimulants just as robust, or do you get reports of more treatment failures or more difficulties in that area?
So we haven't looked at that systematically, but we have thought about two ways to look at that. Obviously, one of them is to talk to the clinicians. We've no evidence. I often talk to clinicians who've worked with these children, and they always say that there's no difference in the clinical response, particularly to the stimulants. No difference, just as effective. However, that's just anecdotal. The other way, of course, is to do some PET imaging studies, looking at dopamine function, for instance. But, that's down the line a little bit for us. Yeah.
Thank you both for some elegant and translational talks. I learned a lot. I'd like you both to talk a little bit about gene-environment interaction, gene-environment correlation. Anita, that we know there's a gap between the SNP heritability of ADHD and its epidemiology in twin studies, and we've been trying to explain that, and maybe the environment plays a role in terms of interaction. Edmund, we know from adoption studies around the world that adoptees are not a typical population, and in fact have high rates of ADHD. Yeah. And to what degree, what do you know about your adoptees, and to what degree does environment interaction with genes play a role in your findings?
Okay. Shall I go first? So actually, Steve, we have looked at that in the past in an old-fashioned candidate gene approach. And we've shown, I think the paper's published in your journal, actually, Steve. Or I shouldn't say your journal, but you know what I mean. We've shown that dopamine transporter genotype moderates the impact of deprivation on the expression of symptoms of inattention overactivity. So those individuals who have the risk allele and the risk environment have the high levels of inattention overactivity, and the other groups within your classical design, they don't have elevated levels at all. So it's quite clear that genes play a role either in making vulnerable or protecting, making resilient the individuals within our sample. So we are looking at that. And the other thing is we are, as I mentioned before, we are doing genome-wide scans, deriving polygenic scores, and going to look at it in a bit more of a robust way in the next month or so. Yeah. Anita.
I suppose I want to say a couple of things. The first thing relates to gene-environment correlation. I could have given a whole talk on that, and I didn't cover that. And I touched on it in relation to the prenatal risk factors, where some of the environmental exposures that we think may be causal are actually correlated with the genotype. So this is a really a big complexity for us in psychiatry, because the sorts of risk factors we think might be important are the very ones that are correlated with genetic risk. So we do need these sort of designs that pull them apart. So let me give you an example that gene-environment correlation is important beyond the prenatal world for ADHD. And these are my kind of favorite findings because you've got convergence from other studies. It's not just genetics. So there was a study I was very lucky to work with my colleague, Gordon Harold, who's looking at children who were adopted away at birth. In fact, it's the EGAD study from the US. What he found is that children at high risk, that based on their parents' ADHD risk scores, their biological parents' risk scores, the biological parents' ADHD phenotype predicted early temperament, which then, in the adoptive parents, predicted mother-child hostility. So it was the kind of innate disposition that was having an impact on an environment and then, which had then further outcomes. And that's also been shown in twin studies where genetic risk for ADHD then results in mother-child conflict. And I think there's medication studies that have also shown that when you treat the young person, the symptoms reduce. Guess what? Mother-child hostility reduces. So again, I think this is the phenomenon of gene-environment correlation is a complexity, but it's also a helpful way of conceptualizing is when you see environmental adversity, be careful about assuming that's what's caused ADHD as a consequence. The issue of gene-environment interaction is difficult, and I would be cautious here. We're still at the stage of discovering genes. There's lots and lots of those genes. We're at the stage still of actually trying to identify causal environmental risk factors, and many of those are correlated with genes. They're not distinct. So I think that whilst I'm a fan of gene-environment interaction, I think you can test it really nicely in animal models where you can knock out specific genes. It's clearly important, but I do not think yet we're really at the stage of being able to look at it really as comprehensively as we would want to and be really confident that this means that if you've got these genes, this is what's going to happen. However, I have an open mind, but at this point in time, I remain cautious, but hopeful.
Good.
Chris Kowalski, stunning study. I have just a couple quick questions. One is, given that the environment was in a high affective load outside of the institution because it was a turnover of regimes, just similarly in wartime settings or turnover, and I'm wondering how much effect was on the children from any PTSD, breaking out that data or information, and how that may affect, because I'm sure that the brain studies have shown some of the changes in the hippocampus and other areas of the brain. My other question, real quick, is that how much of these time was spent with these kids in terms of therapy in England, if they had it, and how did that affect the outcome of the studies in terms of their,
So PTSD, I think one of our children, young people, has a diagnosis of PTSD, one, because of course it's happening very early in their lives. I've never heard that question. I've heard a lot of questions on this study. I never heard that question. There's a very interesting question about the social context in Romania at the time. It's very interesting.
Right.
I think you've got to remember they had very little interaction with the staff at all. The staff basically fed them and washed them with a hose very often.
The reason why I was asking is that I'm retired from the military and I had just a few experiences, and so I got to see a lot of the local area in the Anbar province of Iraq, and so a lot of the kids were coming to us by their families, and their buildings were pretty much not in a nice condition, and a lot of attention wasn't spent with the kids. They were pretty much in these particular areas kind of left to their own on the streets and all. And so I'm just wondering, it's hard to study that, but how much of effect does trauma have on the brain and with breaking out ADHD in terms of the study showing without trauma So that's all.
Yeah.
So I'm going to recommend that we move to the next question, and since you don't have more to say about it necessarily.
I do, but I'm happy to
Well,
hear it.
then you'll continue afterwards.
We'll talk, have a coffee, yeah.
I thought because you hadn't studied it, you would...
I hope you don't mind a layperson's question.
Comment.
Thank you very much for the research and the very clear presentation. I just had one question. I'm wondering what factors might account for children being in an orphanage less than 6 months and more than 6 months. I'm just a little bit familiar with the Ceausescu regime. I know they wanted to have a lot of children, so people were trying to find ways to get out of being pregnant all the time. And I'm wondering if the parents with ADHD were not as good with managing those kind of things. I know there was a lot of alcoholism too among these parents. So and I'm wondering if maybe were they in a different geographic region where maybe they were more appealing as adoptees, you know, or what factors accounts for that?
Yeah, I think it's a great study, but we have a lot of limitations.
Yeah.
We don't know much about that in terms of systematic factors that determine the choice of children to adopt. We know that they pretty much all went in the first couple of weeks, so that wasn't really an issue. And we know they were pretty much all adopted at the time of the fall, around the time of the fall of the regime. So that was a historical event, but clearly there was still some selectivity. But I think that selectivity works against the idea, actually, that the more deprived were the higher risk kids. Because in a sense, the disorder would be more manifest by that time, and they probably would have been avoided if they were showing more risk. Whereas within the under 6 months, it's very hard to see risk for developmental disorders. So I think it works in the opposite direction. So we may be underestimating the effects of extent of deprivation, I think. But we are going to be looking, for instance, at genetic. We know nothing about exposures prenatally, which is a big problem, and there were probably elevated levels of risk in relation to those exposures, but not to produce an 8-fold increase. I mean, in the literature, there are very small effects of these prenatal exposures, and they may not even be causal. So, an 8-fold increase that we've seen in our sample. Yeah.
Thank you.
Last question, Rick.
Yeah, great presentations. I do a lot of disaster work and have over the years, and a couple comments. One is I have a couple of people who actually were in concentration camps in World War II that I still see, and some of them actually have ADHD as well and developed it. But as we look at disasters around the world, and there are a lot of them, particularly a thing like earthquake, like that happened in Haiti, where you get literally hundreds of thousands of deaths, in Haiti after the earthquake, a lot of orphanages, things like that. It would be great if you can come up with ways to predict early on, and should we use, for example, some of the genetic screening to take a look at larger sample sizes to further evaluate that. And there definitely is a lot of overlap, obviously, with PTSD and depression, things like that too, but it would be great to look at larger sample sizes that way.
I guess your question relates to the whole issue of resilience. And
Right.
so we do have about 7 or 8 young people exposed to the really most extreme deprivation in our sample who are totally indistinguishable from their peers in the normal classroom. Some have had, 1 or 2 have achieved wonderful things. And one of them went to Cambridge, for instance. So there are clearly resilience factors within a small group of individuals that can withstand even these sort of exposures.
And that's obviously what we need to work on and figure out ways to do that, because if you look around the world, there are unfortunately way too many opportunities to have these stressors. But great presentations.
Could I say one more thing, Jeff?
Sure.
So, please, can I? Yeah. So, just exactly.
Got to let you talk.
Exactly.
Not too long.
No, very quick. Very quick, sorry. So we are looking at post-adoption factors that can create resilience as well in terms of the kind of bouncing back from the initial problems. And what we're finding, quite clear evidence now, is that establishing a really good relationship, you could call it a selective attachment with their adoptive parent, is a really important protective factor in these young people. Around half of our children manage to do that, and their outcomes are much better.
Absolutely.
Maybe gene-environment correlations, but yeah.
Thanks again.
So look, I told you this was going to be fantastic. It did not disappoint. And I can tell you that the next one is going to be equally fantastic. So we'll have a break for a half hour. We'll come back at 5 after 11 to make up for the late start. We're going to have that one have its full amount of time, and you know, you're going to find that equally compelling.
Interesting data.
We can't really give a bad talk on Romanians.